In this video, we will explain the concept of DNA replication, a type of cell division. Cell division is what allows the replication of our DNA. It's how your body heals itself after getting injured and is also responsible for your physical growth. The way it does this is through a process called the cell cycle. The cell cycle contains 6 steps: the interphase, prophase, metaphase, anaphase, telophase, and finally cytokinesis. Once these steps have been completed, the end result is 2 identical cells. Key terms: prophase, the first stage of cell division. Nuclear membrane disintegrates and strands of chromatin coil up to create chromosomes. Telophase, complete sets of chromosomes are at each pole. The nuclear membrane starts to form around the chromosomes. S stands for synthesis. DNA replication occurs once the replication of chromosomes occur. There is no turning back in the division. This process results into one new chromosome with 2 copies. Sister chromatid, the halves of duplicated chromosome that are held together by a centromere. Centrioles/centrosomes, microtubules found in pairs and move towards the poles, the opposite ends of the cell, when division occurs. Chromatin, distributed in the nucleus and condenses to form chromosomes for cell division. Metaphase, chromosomes line up on the equator, the metaphase plate. Spindle fibers, which come from the centrioles, attach to each chromatid at the centromere. Cytokinesis, when the cytoplasm divides in animal cells, the membrane pinches closed. In plant cells, a plate forms.
We will now discuss the phases in cell division. The first is interphase. Interphase is the process that must occur within the cell before the process of cell division begins. During this step, the cell checks for any damage to the DNA, as well as making sure that the conditions are safe for DNA replication. There is no way for the cell to stop this process once replication begins. Once the cell deems the conditions are proper, it begins to enlarge and generate the organelles that are responsible for mitosis. Once preparation is complete, the cell enters the prophase. Prophase. Inside the nucleus of the cell, there are long, stringy forms of DNA known as chromatin. They stay in this form during normal cell processes. When the prophase begins, the chromatin supercoil into condensed rod-like structures known as chromosomes. Outside of the nucleus, 2 organelles called centrioles move to opposite sides of the cell. In this process, the nucleoli disappear from view and the nuclear membrane disintegrates. The cell then moves on to the next step, the metaphase. Metaphase. During this process, the chromosomes that were formed during the prophase line up along the equator of the cell, and spindly fibers from the centrioles attach themselves to each chromatid, which is 1/2 of a chromosome, at the centromere, center of chromosome. The cell then moves into the anaphase.
Anaphase. The chromatids are then pulled at their centromeres by the centriole spindly fibers to opposite sides of the cell. Chromatids that made up the same chromosomes are known as sister chromatids. The cell then moves into telophase once the chromatids reach opposite poles of the cell. Telophase. The telophase is one of the final steps of the cell cycle. During this process, the hard chromatids uncoil back into chromosomes and the cell membrane begins to form around the chromosomes at each pole. Cytokinesis. Cytokinesis is the final phase of cell division. Cytoplasm divides and the cell membrane closes around each newly formed cell. The result is 2 daughter cells, both exact copies of one another.